student and my dear friends in the Sanjini Mahavidyalaya Sapuli online video class. So student, we are continuously arranging such video lectures for you, and we hope that you all the students are watching and studying such videos. Today we are going to study the another genetic material, RNA. It means that the discovery of RNA as a viral genetic materials, and with the help of evidence, one experiment, that is the Gerer and Schramm experiment on TMV, tobacco mosaic virus. This is the experimentally the Gerer and Schramm, the scientists, they proved that RNA is a genetic material. So most of the viruses, they contain RNA compared to DNA, and in some kinds of RNA viruses, they also contain DNA. But in this case, tobacco mosaic virus is a selected microorganism, experimental virus which contain RNA. And in first time in 1956, they discovered such experiment. They discovered RNA is a genetic materials. So friends, student, as you know that viruses, they generally require a specific host for living. Without that, they are not living. Mostly that's why they are classified. When viruses, they are living within the plant cells, they are called as plant viruses. When viruses, they are living within the animals, that's why that type of viruses are called as animal viruses. When the viruses, they are living within the bacteria, they are called as bacteriophages or bacterial viruses. When the viruses, they are living in the human host, they are called as human viruses. It means that each and every type of viruses, they require a specific host for living. Without that, they are not living. Without that means outside the host, they are not living. In this case, the experiment on tobacco plant and the virus, that is the tobacco mosaic virus. So tobacco mosaic virus generally contain what? They contain RNA. In this experiment, what the important tool is the RNA. TME infects tobacco. TME means the scientists, they observed that on the leaf and leaves of tobacco mosaic viruses, there is the infection, there is the round spherical brown like or sometimes green like structures present on the leaves, and it means that it is one of the infection, it is one of the viral infections. And he observed that tobacco mosaic virus, it infects tobacco, causing the infected regions on leaves to become decolorized and blistered. It means that he observed the specific different types of colorations, round spherical coloration on the leaf of tobacco. And the different strains of TMV produce clearly different inherited lesions on the infected leaves. The scientists, they also observed that what? There are different coloring regions on the leaves. It indicates that the different strains, it means that there are different strains of tobacco mosaic virus. They generally infect to the leaf or leaves of the tobacco. The common viruses produces a green mosaic disease, but a different strain, that is the variant Holmes ribgrass strain, produces ring spot lesions. It means that the amino acid composition of the proteins of these two strains are different. It means that because when the scientists, they observed the protein nature of the viruses, each protein is made up of amino acid. It is the chain of amino acid, right? And when he observes the amino acids in the composition of one protein, it is differ than that of the another. It means that they clearly identify, they clearly discovered what? Two different strains. They affect on the plant of tobacco. So the scientist Alfred Guerra and Scram, they generally isolated RNA from tobacco mosaic virus. How they isolated? It is the process. It is the laboratory process. It is the laboratory process. In the laboratory process, whatever the sample of tobacco mosaic viruses collected in the test tubes, then by using a specific isolation method, for example centrifugation or any solvent system centrifugation process, what there is the breakages of such viruses, what happens, due to which the different particles they are separated, and RNA, that type RNA is separated from tobacco mosaic virus. And when this purified RNA was applied, it means that once they separated RNA from that viruses, and again by that purified RNA, when he applied to the plant tissue, to the healthy leaf of tobacco, the plants develop the same type of lesions that on the leaves they were exposed to intact TMVs. It means that when he isolated what RNA from tobacco mosaic virus, and again that isolated RNA when he affected on the healthy leaves of the tobacco, after a period of times, clearly same symptoms as like a tobacco mosaic viruses previously infected to the plants, that type of infection observed. He said that yes, this is the infections only due to the RNA, because RNA is a genetic material which is responsible for transformation. Transformation means whatever the actions on the leaves or leaf, it is due to the presence of that type of RNA. Guerra and Scram correctly concluded that viral genome of TME is composed of RNA, and he further confirmed that TME uses RNA as its genetic material, and he further carried some experiment on the basis of different strains. He observed two strains he collected initially in this experiment. One is the wild type of strain, and second one is the mutant TME strain. So, and due to the action of these two strains on healthy leaves, what happens? There is he clearly identify, the scientists, they clearly identify two different indication, two different symptoms are observed. One, it looks like green coloration spots, that is the ring like structures on the leaves, and second, it is also a brown coloration with dark spots inside that ring on the leaf of tobacco plant. And with the help of this discovery, he clearly identified, said that this is these are the two different strains, two different RNAs are responsible for infections. Now friends, this is the experimentally he shown, practically he shown that how these two strains are affected. You just see, this is the experimentally how they proved. These are the two different types of TMV. Type A TME, it looks like red color, and second one, type B TME, it is a blue colors. So the Guerra and Scram, what first they degrades both types of TME. Both degrades means he generally centrifuged the samples of type A and type B separately into test tubes, due to which what happens in the to yield RNA and code protein separated. It means that because RNA, they are generally made up of two. One is the genetic material, that is the RNA, or second one is the proteins. The proteins and RNA are separated by using a centrifugation process, and in the next time what happens, in the next stage, the scientists Guerra and Scram, they rechanged the proteins. Rechanged the proteins means the protein of type A TME added into the type B TMB, and the protein of type B is added into the RNA of type A. Means RNA are fixed. RNA type A is in one test tubes, and RNA of B is as it is remains. Only what proteins they are exchanged, and after exchanging what happens? There is the formation of two hybrid TME, that is the hybrid TME1 and hybrid TME2, that two creates hybrid viruses. After that what happens? After production of these two, again he used what? The second, you just see, this is the second figure. This is the second type A and type B. There is the exchange of the proteins, and after exchanging, these hybrid TME further added on the surface of tobacco plants, which healthy tobacco leaves. What happens? When he inserted these samples, these culture of viruses on healthy leaves of the tobacco, after a period of times, what happens? He clearly identify what? There is the symptoms. There is the symptoms of tobacco mosaic virus, but after that he again collected the samples from that leaves. It means that whatever the spots are observed on the leaves, such spots are separated means he researched in the laboratory level, and he observed that whatever the type A progeny, it generally looks like TMB, and type B progeny, it further looks like TMEB. It means that whenever we changed the proteins, there is no change in the progeny or in the viruses, because RNA is a genetic material. Whenever we change the proteins, there is no change, because RNA is remains. That remain RNA generally produces same type of progeny, and with the help of this experiment he proved that yes, what? RNA is a genetic materials. This is also one of the figure. In this figure, same type A, type B, separation of proteins. There is the hybrid TME produces. Yes, these TME, hybrid TME further, when we added on the leaves, healthy leaves of the tobacco, what? There is the production of that infections on the leaves, and that infected leaves, when we again what observes from that spots, when the spots generally removed in the laboratory levels, and on that spots when we observed which type of progeny is present, that times they observed that what? The RNA remains constant, RNA remain present. Whatever the changes, there is no any problems on TME. And with the help of this experiment, the scientist Greer and Scram, he proved that RNA is a genetic material. RNA is a viral genetic material, and which is responsible for heredity character, which is responsible for transformation from one generation to another generation. It means that there is no any other part like protein. It is not a heredity character. With the help of this experiment, they proved. So friends and my dear students, so with this experiment, you clearly identify, you clearly studied how the RNA is a genetic materials. So friend, you enjoy, watch the video, and side by side you learn, you study the notes as well. And on the internet you have lots, you have available the different theoretical as well as the different material on this subject. You study well, and if you have any problems about this topic, You ask me, I am always ready for answering your questions. Thank you.